Right, so I think we can start on time. And our next speaker is James Glazier. There's a title change. He's going to talk to us about making virtual tissue modeling an integral to biology. Yeah.
Okay, I was up all last night rewriting this talk, which is always a dangerous thing to do, and so I apologize in advance. I also apologize in advance because the talk is going to be long, and I'm going to go through it fairly quickly. But if people are interested in the details, you'll have it online and you could go reference them. There are an almost limitless number of people who participated in this project over the years. I want to single out Giulio Belmonte, who's now at EMBO, who's done a lot of the modeling I'll talk about, Sherry Clendenin, Abbas, who's now at the St. Jude's Cancer Hospital as a faculty member, Andre Samoji and Maciek Swat, who are two of the lead developers. I'll show some unpublished work by Gilberto Tomas and, boy, I don't have her here, Rita de Almeida. She's hiding in here somewhere. I should have highlighted her. And then some ideas that were contributed by James Sharp, Octavian, Francois Grenier, and some others. And I'm sure there are people I've left off. So if you're in the audience and you say, I left somebody off, please tell me and I'll add you. Some of the funding sources over the year, primarily NIH, more recently Falk Medical, and the National Science Foundation and the EPA. I want to start out with just two very simple definitions. When I talk about a model is an abstraction that embodies some set of hypotheses and components to be tested. When I talk about a simulation, I'm going to be talking about a computer program that contains a model implemented as a set of methods to numerically evaluate a particular instance of the model. And there are many, many layers of abstraction between models and simulation, and one could easily spend a week of lectures just talking about how to go from here to here. When I talk about a workflow, I'm going to be talking about what several of the people yesterday, we heard in the morning about several workflows. Philip Mades talked about them. Eric Sahai talked about them. The whole process of going from biological experiment to model to simulation to data to comparison with experiment and back. That's a workflow. And again, to make this workflow into the actual detailed process of doing science would take a week of lectures. It's very complex. Every model, regardless of whether it's a diagram in a textbook, a mathematical model, or anything else, has four classes of components. It has the objects that are being modeled, the description of those objects' properties, behaviors, and interactions, the dynamics by which that set of objects and processes evolves, and subset of initial conditions. That's true for any model of any level of detail. You could always make this kind of breakdown. Why is bioinformatics, and we're not bioinformaticians here, I don't think. If you're a bioinformatician, raise your hand. Okay, you see none. So useful. There are 100,000, probably to 1 million people who use bioinformatics tools every day in the world to do their science. It's inconceivable you could have modern biology without bioinformatics. It's totally integrated into the process of modern biology. The people who use bioinformatics don't care about the methods they're using. They care about the results. Why is bioinformatics successful? First, the tools provide information that people find useful or essential. Secondly, the tools are integrated with experimental workflows for modern biochemistry and molecular biology. Third, they support the whole scientific workflow from literature review to hypothesis development to experiment design, experiment execution, analysis, data archiving, and knowledge capture. They're tools for all of those things, and they're standards-based, which means that they all connect to each other. All the tools use the same standards for data, for models, and for software interchange. So they all work hooked together without having to worry about it. No adapter plugs. The software and methods are modular. So if a new, better method comes along, or you want to check whether there's a method-dependent effect, you can simply change that particular component in this whole ecology. And lastly, the tools use biologically natural representations of data. You have concepts that are built into the tools of things like genes, pathways, regulation. These are natural concepts, and when you work with those tools, you describe the things you're working with in terms of biologically natural representations. Now, we could come up with a lot of additional reasons bioinformatics is successful, but these are just some. And so if we want to be successful, we have to do the same thing. It's not easy. Okay. Here's a typical modern virtual tissue model containing all sorts of components of different scales, integrated to build some kind of simulation. Here's a little toy cancer simulation we did a long time ago of vascular tumor growth and the effect of neovascularization on tumor growth. Here's me, Chicken Little, saying the sky is falling. But this is really a lecture for the students. The people who are in my era are too old to change our ways. Who said, was it Philip that said that science proceeds by funerals? So I'm hoping that maybe I can help the students avoid some of the mistakes that the older people have made. So this is an example of do as I say, not as I do. Okay? We believe, I think all of us are here because we believe that mathematical and computational biology have the ability to transform biology, to transform our understanding of biology. Some of us have committed our whole careers to that. And yet, I just got an NIH report written by some very famous people that says, quote, the user community, referring to all mathematical biology except bioinformatics, remains small with so far limited impact in terms of publications and those almost exclusively in the modeling literature. Hardly any published work in the primary biomedical research literature has yet arisen. Now, we could all say that that's totally unfair, and we could all cite some counterexamples. However, this is the official view from NIH. Yes?
So is that a criticism of the modeling community or those who go to the primary literature?
This is a criticism saying that you should shut down the entire modeling effort at NIH. This is what the biological world thinks of us.
Is this limited to the issue of virtual tissue modeling or methodology and its entirety?
This is basically talking about all of the things that we would call mathematical and computational biology. As I say, bioinformatics is really a separate field, and it's not what they're talking about. What we would call predictive quantitative modeling is what they're talking about. And when I say virtual tissues, I'm using it loosely because there is no good term for what we do. So I'm adopting that. Okay. And so to be accepted as a normal part of biological science, we're going to have to do more than just develop methodologies. To be productive as science, you have to do a couple of things. And this is, I hope, something that in Europe people are taught when they're students, scientific method. In the United States, people assume scientific method, and therefore they don't teach it, and they don't know it. We've heard a very good talk by Dr. Uberlis yesterday about the issue of verifiability and falsifiability. And that's certainly important. There are two other things that I think are maybe less well understood or thought about, one of which is reproducibility, which is something every experimentalist in this meeting has talked about in great detail. And I'll go into a little more detail about that. And the other one of which is extensibility. That is, science has to proceed by the accumulation of knowledge. It's progressive. And I think we often don't take that seriously enough, although many of the talks here, at least some of the talks here, have addressed that in some specificity. And I would say that our key need in some ways is to make this general area of mathematical and computational biology more scientific. What does that mean? Everyone says, well, I'm doing science already. Well, most modeling results, as published, can't be reproduced at all, or at least not without enormous effort. Even when people publish simulation code, you can't extend it. It's not written to be extended. Methodologies are not easily combinable. Models are not designed so that you can reconfigure them, extract components, modify them, and so on. And most model components are not designed to allow independent validation. And that's a critical engineering criterion which we don't tend to pay attention to. All of these things are best practice standards in all sorts of areas of engineering and science. However, we don't always follow them in our area, and I'm afraid we often don't teach them to our students. As a sort of a simple example, there is an accepted standard for specifying biochemical reaction networks called SBML, and I'm sure a lot of people who've given talks about reaction kinetics in this series have used SBML, but I haven't seen the word once. Not once. If you're using Mathematica or MATLAB to solve your coupled ODEs instead of SBML tools, you're fighting all of that. You're basically holding things back. And you have all sorts of good arguments about why SBML tools aren't so great. Nevertheless, if what you want is for your work to be a foundation for somebody else and for advancement of science as a whole, rather than just for yourself, you make do with the standards. Okay. There is a specific technical issue about reproducibility, and I went back and forth putting in slides and taking them out because, again, you could lecture for a week just on these issues. Replicability is what you get if you publish your code. Somebody can run exactly the same code, if they're lucky, often they can't, and check that it does what it says it does. That doesn't give you any understanding about how it works. And that's not the primary standard in science. Reproducibility means that you can confirm original results using independent methods. That is, you can check that your results represent reality in some sense rather than artifact methodology. Reproducibility is a cornerstone of scientific methods. Experiment, in general, modulo the occasional scandal, we don't accept scientific findings until it's been reproduced. That doesn't mean replicated. It's not true?
No. You know the recent scandal about what? A very high percentage of results that appear in the literature are not reproducible?
Exactly. And those tend to disappear. You hope they disappear if they're not right. If you can't reproduce a result, it's not a scientific result. Until it's been reproduced, it's not there. That's my point. The vast majority of computational biology work is never reproduced. That's true of experiment too, but it's even more true of computation. And it should be easier to reproduce computation than experiment. Okay.
Well, that is also not entirely true. I mean, it is very difficult to go and, as a modeler, what you do is you don't necessarily reproduce somebody else's result in the context of their model. You reproduce it in the context of your own work, and finding the same result, at least in the right range of parameters, in the right limits, convinces you that what other people did is correct. You don't necessarily reproduce their own calculation, because that would take the life of a PhD student yet again.
What I'm trying to say is, in a sense, agreeing with you. That is, that what we need to do is to make it so that it is not a PhD's worth of work to reproduce a result. And until we do that, we are not going to be taken seriously by biologists. And again, you'll find some wonderful biologists who will take you seriously. But the whole biological world will follow that little quote that I gave you from NIH. Okay. We've just discussed why reproducibility is important, and I'll point out that if you can't reproduce, you can't validate, and you certainly can't then use your results in critical applications like clinical settings or regulatory applications, which are two of the biggest places that need what we do. And it's also very inefficient, because when you write models and simulations, all the information you put into those is essentially lost. It has to be reinvented every time one wants to use it. And that means everybody has to duplicate effort, and it makes progress very slow compared to what it could be. Again, look at bioinformatics. Yes?
By losing the knowledge, do you mean the sort of thinking process that you go through in throwing some things out and keeping some things in?
That's right.
If you present it with the equations, you have the model.
No, you don't. The equations do not embody the abstractions they're derived from.
All this program does not depend on scientists. It is a matter of funding policy, of publication policy. Under circumstances that we have, we cannot work in this way, unfortunately.
I'm happy to make excuses for my failures, and we can always, over coffee or better over a drink, lament the reality of the world. I'm laying out a program for where we have to get if we're going to be taken seriously. We could say it's not possible to do that. As my thesis advisor used to say, physicists are poets. The world doesn't need very many poets, and the world certainly doesn't need mediocre poets. And so for those of us like me, who are never going to be Stephen Hawking, one of the things I am concerned about is to make the work I do useful. I like to do new science. I like to do things that are new and exciting. But I also care about use. So in that sense, I'm more of an engineer, perhaps. And I completely agree with you that there are many institutional blockings for this. But again, the students should at least know what this cloud cuckoo land of Aristophanes is, that we can then come back to. Okay, so how can we make things useful? Well, tools should give actionable results. That is, they should support the entire workflow of model development, execution, validation, everything I showed you before. Our current tools support only a tiny part of that. If I go from experiment, biological observation, extraction, hypothesis generation, there are a whole set of tools we need there. To go from there to a biological model, we need tools. To go from there to a mathematical model, we need tools. To go from the subcomponent models to integrate them into a multi-scale model, we need tools. Once we have that, we have to instantiate it as a simulation, and then we have to run it. And 95% of the effort in this meeting, which is fine, has been talking about solvers, which are just this little piece in this corner. Once you have simulation output, you have to analyze it, process it, organize it, archive it, and the talk yesterday by David talked about some of the issues about how you compare experimental data to simulations and use it to optimize simulations. And then you need tools to design experiments. Again, David was talking about that a little bit. And then finally, you need tools to capture all this knowledge and organize it and present it in ways that are useful. And all I'm saying is if you don't have this whole ecosystem, no matter how much you work here, you're not going to facilitate this whole workflow because the bottleneck is over here. We all know about limiting rate reactions. Limiting rate reaction isn't down here, it's elsewhere in this pathway. Okay. Modeling tools need to be usable. That doesn't mean that developing new numerical methods isn't very important. It's extremely important because if you don't have new mathematical numerical methods, you don't make progress in your ability to solve hard problems. However, if you look at science, if you look at computing in general, software development, and it's certainly true based on my own experience, 20 plus years doing this stuff, algorithm development is much less than 1% of the development effort of a substantial tool. In other words, developing these beautiful meshing algorithms and so on is somewhere around 1% of the effort to actually develop a tool somebody can use. Turning that into a computing instantiation is another couple of percent. And then wrapping that computational engine in something that makes it usable is 98% of the effort. Now you could quibble about this and say this should be 3%, this should be 7%, this should be 90%, but the ratio is 10 to 1 to 100 to 1. There's not a lot of complex mathematics or even numerics in most of the tools we use every day computationally. Usability is the key. 3, we have to build models using abstractions and workflows that are appropriate to the people who use them. It's always going to be hard to understand biology. If somebody does the hard work of understanding biology, it shouldn't be hard for them to implement that as a model or a simulation. It's our responsibility to make that easy. Suppose when you used your word processor, you had to enter your English in Japanese characters, which were then translated back into English. It would be a perfectly good word processor. Nobody would use it. You'd use a typewriter or a piece of paper. That's where we are in our tools. Okay. Who are the people that matter? Biologists. 99% of the world, biologists, mathematicians, physicists, and everybody else, this. Okay. That means we have to make our concepts appropriate to them. And that means if you develop a modeling approach, it has to be presented in terms of biological rather than mathematical and computational abstractions. This actually makes a lot of sense because the results of a given biological situation shouldn't depend on the solution method that I use. And I wrote here this wonderful ordinary differential, this definite integral. Pi equals the integral from 0 to 2 pi sine squared theta d theta. This has a definite meaning, and whatever numerical integrator I use, I should get the same answer within some level of accuracy. The truth of this statement shouldn't depend on whether I use a particular numerical integrator. And the way I frame this statement shouldn't make me say, well, I'm going to use stochastic integrator, and therefore I have to write this integral in a different way and have to think about the integration in a different way. So we can't ask biologists to think about things in a different way. Biology is full of modular concepts: individuals, organs, tissue, cells, molecules, behaviors, division, growth, circulation, interactions, chemotaxis, initial conditions. You have to be able to specify models in terms of biological concepts. If you can't, You can't identify your model hypotheses. That's why a set of PDEs or ODEs does not allow you to invert to go back to the underlying biology. You can't reuse them. That means you're not cumulative. You can't build libraries, cumulativity. You can't connect them.
Question?
Yeah.
I don't understand your statement about given the PDE, I can't go back to the biology. Of course, I don't have all of the biology in, but I have identified some processes that
Yes.
by some rationale are important, and I put them into the equation.
That's true, but the correct way to do that would be to have metadata annotations that document what the underlying biological concept was and allow you to invert. If you do, let's take, as for us old-fashioned people, Fortran. When you do Fortran compilation, you have two options. You can do compilation with symbol tables, in which case when the thing crashes it tells you variable such and such, x was equal to 0 when you divided by 0 on line 57, or you can turn that off, in which case you get a core dump. I'm just saying that if you don't keep that reversibility, you keep the higher level abstractions present, it makes it very difficult for people to go backwards. You have to guess what was intended from the equations rather than mapping back and forth in a clear way. And there is a formal way to connect a pathway diagram to a set of equations in a way that you can go back and forth transparently.
But even in that context, you're typically not going to be able to describe all the components in the pathway. I mean, we saw numerous examples of these very complicated biological pathways. You're certainly not going to be able to include all of that.
Of course.
Even if you could, you don't have the data required to say.
That's fine. This is all fine. Nevertheless, you should have a diagram that shows the pathway you are modeling and what your assumptions are. If you look at, for example, JDesigner, which is a nice tool to build SPML models, you draw a pathway diagram, you identify your components, and then, once you've built the structure, the model structure to biological level, you annotate that diagram with the mathematics that turns it into a mathematical model. That way, both the picture, the biological conceptualization, and the mathematics are all kept together and can be pulled back and forth any way you like. I'm not saying you can't learn to do it in your head. I'm saying that your users aren't going to, if you have to do that, they're not going to use it. If the only way we could organize DNA sequences was in 1,000 base pair blocks, and we didn't have genes, pathways, and so on, I promise you bioinformatics would not be where it is. That's the problem. Okay. Another thing, and this comes back specifically to what Hans is talking about, and it's a pain, but critical, is that you should be separating the biological structure of models from the implementation. Most modeling methods mix description of the model biology with the solution methodology. And that has all sorts of problems. This is the one we were talking about just now. It means that I can't easily say, I've solved this integral using a stochastic integrator, now I want to solve it using a Cauchy integrator. Can't do that. That means that I can't reproduce easily, and it inhibits model reuse, extraction, and so on. This is as if each DNA sequencing method worked only on one gene in one organism. And so we had to reinvent sequencing and build all of our tools again each time we wanted to look at a different gene in the same organism, or the same gene in a different organism. Or maybe another example. If you had to rewrite Microsoft Word from scratch every time you wanted to write a new letter, you wouldn't write a lot of letters using Microsoft Word. Maybe people don't use Word. Okay. As an example, if you have cells that are doing things, we have many ways of representing cells. You could represent them using cellular automata. You could represent them with rule-based methods. You could represent them as point objects interacting with potentials. You could represent them as clusters of point objects interacting with potentials. You could represent them using immersed boundary methods. You could represent them using finite element boundaries. And we've actually seen, I think, examples of almost all of these in the lectures we've heard. Some of them are more useful for certain kinds of things than others. Or we could do these POTS-type simulations, which Luigi Preziosi and Marco Scianna have written this great book on, so I recommend that. This is the method I use because it's a cheapo method. It's not very accurate, it's sort of simplistic, it has a lot of problems, but does allow you to add any conservation laws or physics that you want in the form of constraints. It's very flexible. However, if the underlying biology is the same, whatever solution method, just like my ODE, my integral here, whatever solution method I have should give me the same result. If there are differences between the results using different solvers for the same biological model, and that biological model has already had all the abstractions and simplifications that Hans points out we need to make, I'm not saying you put everything in. You throw out things, you reduce things, you define it as a biological model in the reduced form. Any differences between those results don't represent the biology, they represent artifacts of the solvers you're using. Generally, as Hans points out, higher accuracy methods aren't really the issue because our uncertainty about the biology and approximations are so big that those errors dominate the numerical errors. Not true if you have numerical instabilities, of course. And it winds up that very often you can cheat because biology is noisy and robust, and so you can get away with sort of crummy methods and get something that's not too bad. Okay, next. We can't predict how people will combine model components because biology is too diverse. And that means you have to build methods that are flexible and reconfigurable. Again, and if you're a LaTeX fan, excuse me, if you use a different word processor to write letters and articles, that's not convenient. You should be able to use one word processor to write text. Mixing simulation methodologies together in monolithic code has all sorts of problems. Can't tell if a method does what it says. You can't replace one crummy method like Potts model with a fancy new method like phase field. We can't take out the meshing, fixed meshing of the Potts model and put in the beautiful adaptive meshing we were just seeing. That's what you lose if you're not modular. And that's what engineers would call future-proofing. Since you don't know how things are going to evolve, if there's a technique that could be replaced by something better, you want to make that modular so you can take it out and replace it when there's a better way to do it. Okay? How am I doing on time? Half an hour. I'm not
You've already done half an hour.
Sorry?
You've already done half an hour.
Okay, good. I've already snowed everybody. All right. Imagine if on your phone, every time you want to load a new app, you had to recode the C++ of the operating system. That's where we are with most biomodeling. Again, some people would use it, but I promise you it wouldn't be 4 billion people. Now, the downside of apps is that they have to follow the API that's standardized, that's presented to them, and that limits certain things. It makes them maybe less beautiful, less efficient. But the benefit is that people can interconnect them and use them. And they often interconnect them and use them in ways that the person who wrote the app never thought about. And so if we don't want to be completely solipsistic in our research, In other words, we want to develop things that not only we use, but other people use. We have to accept that people may want to use them in ways we didn't plan on. That's really critical. Okay. Now, why is this so hard? Well, we've seen pictures about multi-scale elements in biology. There are many different techniques at each scale. They often don't couple very well together. We have issues of time scales. We can't model everything. We have to pick ranges of scales. We have to make simplifications. That's pretty hard. There are many other things, and I'm sorry this is so small because this list kept getting larger and larger. Biological terms, this is something that's very important actually. Our last lecture very, very importantly said you have to start with definitions. Winds up that if you look at things like cells or growth or almost any biological concept, there isn't an unambiguous definition. There's almost always an exception in biology. And so then you have to have fuzzy definitions that say this is true most of the time. We don't like that very much when we write computing programs or mathematics. Okay, structures overlap in multiple ways. They may not be hierarchical. Boundaries are fuzzy. Concepts are fuzzy. There are many, many components to worry about. Scaling doesn't work. We have to couple 1D, 2D, and 3D objects. There are things like long-range interactions and inhomogeneous meshes, which are a pain. A lot of the science is still fuzzy. The description of extracellular matrix meshes still not well established. Viscoelasticity, there's no general theory of viscoelasticity. Transport and advection in biology are very complex because you have active and passive transport that interact in complex ways. So there's some basic science that's still not nailed down, and the best we could do is provide placeholders for that, put in the best we can, and hope that somebody comes up with something better to plug in. But we better make sure that we've isolated those things so that when somebody comes up with a better method, we don't have to suddenly rewrite 100 million lines of code. That's as good an example of why we need modularity as anything else. Cross-scale connections are very complex. As Hans likes to point out, it's very hard to come up with parameters values that are reasonable for coarse-grained approximations. The other day we heard about Young's moduli in a particular context. Well, most materials that are biological don't have a Young's modulus because they're viscoelastoplastic. So there is no Young's modulus. The Young's modulus is an abstraction, just the way viscosity is an abstraction, that applies under certain very specific conditions. Those conditions are often not met in biology. On the other hand, the general abstraction may be useful enough that you want to get away with it. But you have to recognize that it's not a complete description of the reality. Okay. The information that we get experimentally, like the level of compound X inside cell Y, often doesn't translate in a simple way to the thing we care about, like how motile the cell is, or how much it sticks to another cell, or how fast it divides. And so there's a big problem, which is how to connect the biochemistry, which we now have a lot of experimental techniques for, to the things that really matter, which are cell and tissue level behaviors, which we have less ability to measure and control, and which are really the important things. And so we do have a bit of the situation in the experimental biology world, and people here have been talking about new experimental methods to get away from this, where people look under the light post for their key, because that's where it's bright, it's where you have the tools, but they don't look at the things that are important over there, because it's dark, it's more work. Okay, and very often the natural biological components in a model may not correspond to the way you would naturally discretize a simulation. And in that case, our responsibility is to develop essentially parsing tools that take a natural biological description and parse it into something that's computationally appropriate. But we need to do that. We can't leave that to the user to do. Not possible. All these workflow tools, standards, and so on are really unglamorous and boring to develop. But that doesn't mean they're not important. how many levels of abstraction are there between your computer and your internet cable? There are at least 9. And those are all based on standards. And without all those 9 levels of standards, you couldn't connect. Yeah?
Not just unglamorous or boring, but it's not supported by any granting mechanism.
Oops, sorry. That was,
Nobody's going to give you lots of money to make a GUI, for instance.
software sustainability is tough. Finding money, finding people. So maybe we have to think about new models for doing this stuff. Yeah.
So your internet example is perfectly fine because the system is well developed and we know all the components, etc., and we know how to set standards at the different levels of communication. But do we even understand a complex biological system like the cell well enough to even think in those terms?
That's a legitimate question. In other words, should we just wait? And the answer is that you have to do both, and that's an example of even more why you need modularity. If you look at the history of internet, for example, IP addresses have been extended recently. That was possible because the people who built the original IP domain structure realized that that might have to be replaced. And so the way you deal with that scientific uncertainty is to design in ways that you can replace concepts as they are developed. And the more homogeneous and integrated your modeling tools are, the harder it is to do that. And that doesn't mean you don't make mistakes and have to go back and redo things. If you look at things like IBM token passing rings and the different standards for cell phones, sometimes you just say, okay, we've learned a lot, now we throw that out and start over. But that shouldn't be something that's done regularly. It certainly shouldn't be something that's done by every group in every paper. It should be something that's done in a considered way and as infrequently as possible. And it's possible to design things in a way that minimizes that. Not eliminate it, but minimize it. The transition from horse-drawn carriages to cars was fundamental. On the other hand, the shape of early cars looked like a horse in front of a car, and the roads were the same. And then they had to invent things like traffic lights. And so you don't necessarily have to wait until you know everything in order to start building tools. In fact, if you wait until you have everything to build the tools, then you may wait forever.
Yes, but this somehow assumes a top-down approach. Bottom-up.
No. Not top-down at all. This is what's actually, and I don't have time to go into it in detail, is what's called community development. Linux is a community development project. Everything that I'm going to be talking about that I believe in is open source. It's less efficient to do things that way.
I think it's top-down approach to the problem where you say, okay, let's say I have this process, this process, this process, this process, this process, and all together this is going to make me a picture, right? And then I can take one of these moduli and put it back in a more refined way. But the problem is that we don't even know what are the moduli at the moment.
That's right, and that's exactly something I'll come to if I have enough time. Yeah.
Vince, could you comment? I'm actually ashamed to admit that I never could understand what's the difference between top-down and bottom-up. I see that everywhere and never could figure out.
It's a loaded term and it doesn't really have a well-defined meaning. Top-down usually means that you pre-specify the architecture that you're going to use and then subdivide it. So top-down is analytic and bottom-up is synthetic. And biology is very much synthetic. You have self-organization of things. And so that is a classic bottom-up situation. On the other hand, a saying having a standards board that says that the British plug will be designed to supply very large-scale heating units, so it has enormous copper connectors, is a top-down decision. To say we're going to drive on the left or the right is a top-down decision. But it tends to be more of a political term than a scientific term. As an example of a successful integration platform, we could talk about MATLAB, which is used by millions of people. It's commercial, which I don't believe in personally. That's why we don't use it, because we believe that things should be free. But the great thing about MATLAB is it has easy-to-use scripting, makes it accessible. You can build toolkits, which can extend it for specific applications. It has a good user interface. It's shareable, can be used in teaching, it's well documented, it has training, it's professionally maintained. Now, why couldn't we just use MATLAB for computational biology? The answer is primarily, one, it's not open source, it's commercial, although that might be a direction to go. And two, the abstractions it uses are fundamentally mathematical abstractions, not biological ones. Okay. If you could make these things work, and you look at your cell phone, and you look at navigation software in your cell phone, just think about all the levels of standards, workflows, and so on that go into your being able to say, I want to go from here to London, and have Google tell you that if you walk from here to the train station, there'll be a train in 20 minutes, and it'll get you to King's Cross in 49 minutes because there's a delay of 30 seconds in station such and such. That's all standards-based, and there is an immense infrastructure that allows that to happen. And it's developed based on interchangeability, heterogeneous data integration, and so on. That's what you can get when you do this right. That's where I think we should be. Do I believe we can get there tomorrow? No. Do I believe we should wait 40 years to try to get there? No. I think we should have a vision of where we need to get. Yeah.
Who's going to decide? Is it going to emerge or is it going to be decided?
Sorry?
How is it going to happen? Is it going to emerge or is it going to be decided by some group and then people follow?
Well, it always works out that it's both, isn't it?
Yeah.
There's a group of people who believe, who come together, who make the effort, and then if that effort is useful and successful, people join in. And there always are going to be some renegades who come up with new things, and eventually those new things usually wind up replacing the old standard. That's good. But you could use sort of an 80-20 rule or a 90-10 rule and say, ideally you have most of the work going on in the mainstream, and then you have some work that's developing what's going to overthrow the mainstream. And we're in the opposite limit. We're in the 95-2% rule, where 95% of the work is going off in all directions, and 2% is trying to build infrastructure. What I would like is to be able to take these kinds of movies from, this one's Nadine Perras, but we saw these beautiful single-cell movies yesterday from David Ubelis. Take that experimental data, digitize it on the fly, build a simulation on the fly, and have a real-time movie of my simulated zebrafish embryo that I could overlay with my experiment as I'm doing it to learn. That's a dream, but it's not an impossibility. We just aren't there. Minimal standards. First, adopt best practice from other fields. Support multiple modeling methodologies. Allow easy import of experimental data. Use scripting languages. Never hard code things for model description. Open source, modular, extensible, cross-platform. Easy to use. Linkable. Community developed and supported. ImageJ, the standard imaging tool. Open source software, all those plugins and things were developed by community. MATLAB, main code is commercial. Many of the toolkits were developed by private researchers who made their tools available to others. Python, open source scripting language. Hundreds of open source packages developed by the community to solve problems they need. Python standard, originally, one person. Once the standard was developed, everything else was done by community. Because ultimately there has to be a standard for these things. Or many standards, Standards evolve, but they have to be thought about. When there are standards, use them. If you're going to talk about biochemical networks, you bloody well better put them in SBML, even though it's annoying. Document to allow third-party development. Develop modeling standards to allow code reuse. Agree on core technology to avoid duplication. Future-proof. Focus on usability. And provide support. All hard to do, hard to fund, different thought process. But again, there are students in the room, I hope, and those students can begin to think about things in a brave new world that's happier than the one we're in. Okay, virtual tissue environments. This is just model solving. These are tools for building simulations. And the one we developed is called CompuCell. There are plenty of other ones, Morpheus, Simune, Chased, Cellsys. We heard about LibCell yesterday. And there's some others. But surprisingly few. And surprisingly few of them meet the criteria that we've just enunciated. And I should be first to say that ours doesn't either. We're always trying to make it better. What is CompuCell? CompuCell is a cross-platform, open-source environment for building and running these multi-scale simulations. It's written in C++. You script in XML and Python. That means that you can describe simulations in 50 lines of code, 100 lines of code, sitting on a million-line code base. It gives you basic things that you might want, like PDE solvers, cell evolution, subcellular network models in biochemistry. It connects them up, doesn't do it in a very sophisticated way. It has graphical user interface for model specification and execution. And it supports some parallel stuff. If you're bored and you're doing your email, you could go to our site, CompuCell3D.org. It's a one-button installer for Windows and Mac for some species of Linux.
Question?
Yes?
So, exactly directed toward this issue you raised earlier, how easy would it be for me to come into CompuCell 3D and change the module for how a cell moves?
You could do it in Python in half an hour, but the execution speed would be slow. It provides support for doing it in C++, where all of the boilerplate is done for you, and that might take you a day. If you wanted to pull out the full PDE solver and plug in a fancy new PDE solver, you would have to think about how to define the abstractions in your PDE solver in terms of the APIs we have, and that's a bigger process. We're in the process of integrating Phoenix into CompuCell. The biggest problem for that has been that the Phoenix people have this ideological hatred of Microsoft, and therefore they refuse to support Windows. And so Bacek has spent a year recoding Phoenix so it'll run on Windows. Because we won't do anything that doesn't run on Linux, Windows, and Mac.
Sorry, what's Phoenix?
Phoenix is the biggest open source PDE solver. Has very, very fancy PDEs, and especially advection diffusion and fluid solvers. It's a beautiful package, but also not easy to use in terms of model definition. So we're trying to write a front end for it. Okay. CompuCell provides abstractions like cells, finite element structures, fields, reaction networks, and so on. And the key thing that it does, more than anything else, is it allows you to describe these things at a fairly high level. It doesn't completely separate model from instantiation, but it makes a stab at that. We're trying to do better. Because I'm running out of time. I have I guess I still have, what, 20 minutes?
20 minutes.
Okay. The great thing about CompuCell is that for a new user, it provides what they call the wizard in its special editor called Twinit. It's still text-based for model specification. But where you type in the name of your model, You define what the objects are going to be in the model. You define the general model parameters. You define what the basic properties and interactions of those objects are, and it builds the model for you automatically. Now, you still have to go in and edit the parameter values and things like that. If you want to do fancy things like have the growth rate of a cell controlled by chemical concentration of CDC whatever, you may have to go in and do some little Python tweaks. But in its essence, you can build a simple model in about 1 minute. So a couple of weeks ago, I went to Cold Spring Harbor. They had a class of experimental biologists, microscopists. None of them had ever tried to build a multi-cell model before. In an hour and a half, we were able to download the software. Everybody was able to install it. We went over a simple model of primary vascular plexus formation by HuVex, wrote down the biology, wrote down an abstract biological model, wrote down a mathematical model, implemented it in CompuCell, ran it, and then we're able to see the effects of parameters on the behavior of the model. And by the end of the hour and a half, there are people saying, well, what if we had 2 diffusing fields, or what if we had this regulator or that regulator? And they were already doing that. These people had never done anything of this kind. That's pretty good. That, to me, was a success. Does it mean it could be easier or better? No, absolutely, it could be much better. However, if you come for a week, at the end of the week, you could write a model of almost arbitrary complexity. And you wouldn't have to learn that much coding. You would have to learn a little bit of Python. We have a 1-day Python boot camp for people who've never done programming before our course. Okay? And what do you come up with? You come up with model descriptions that look like this. This is metadata for the model. This is the cell types, specification of contact energies. We have a diffusing field. It has a name. We have an initial condition specifier, and so on. And then Python does the things that aren't abstractions that we've abstracted yet, because we always have to have a box that says you can put anything in this box. That's how you extend. Okay, and here's a simple simulation. You run it in a little player. You can monitor and steer it as it runs. You can see fields. You can see cells. This is a 2D simulation, but it's intrinsically 3D. And I wanted to show you a couple of applications to just show you the different, why you have to have flexibility as your primary criterion. Because you don't know what, I think Philip was the one who said that you have to expect the biologist to come to you, not the other way around. So that's how we work. We're an enabling tool. I'll give you a toy model because I promised it, and then I'm going to talk a little bit about 1 or 2 other models. So here's a tiny cell crawling model done by the Brazil group, University Federal Rio Grande do Sul, and François Granaire, Paris. That's really a demo more than anything else. We've seen our biology of cell crawling 1 million times. We're going to abstract it in 2 very, very naive ways. We're going to assume that there's some chemical field which is variable within the cell that causes membrane protrusion. And we're going to assume that binding of the cells to the substrate is hard, so there's no advection relative to the substrate. Makes life a lot easier. So we're going to assume that the only transport within the cell is diffusive, and that once a molecule attaches to a stress fiber, it stays put relative to the ground it's sitting on. And again, it's a toy model. We can just we can modify that as well. In this case, our pot cells are turned into multi-layer objects. So there's a nucleus with one set of mechanical properties, a cytoplasm with another, and some representation of a lamellipodium or lamellum third, and a substrate. You can use that same kind of representation to do epithelia. And you have simple mathematical description of this chemical field, force generation, volume conservation, potential cell adhesion if cells can touch each other, and so on. And here's a cell without a symmetry breaking, and it's generating a little bit of leading edge. It's not doing much, fluctuating. And then we have a little symmetry breaking, and our cell starts crawling. It's a crummy model, but it just is a proof of concept. Now you layer in the biology. It's 3D. The nucleus is 3D here, and the cytoplasm here is transparent, which is so, because otherwise you can't see the lamellipodium. You can track the cells. You can compare the displacement squared versus time as a function of model parameters to experiment. So, second example, planar cell polarity. This isn't the cell movement model, and some of this work was done with Octavian Wojciechowski and his people here in Cambridge, mainly Giulio Belmonte's work in EMBO, and a variety of other people, Jim Sharp and his colleagues in Barcelona. Basic question here is that at epithelium, how do you get polarization within the epithelium? In this case, you take your single cell and you include mixing regions within it that represent things like cell adhesion molecules, transmembrane receptors, junctional molecules, and so on. They could be integrins, efferents, cytoplasm, cadherins, and so on. And you allow these things to rearrange based on what they find in their neighborhood. If I take these cells initially that are nonpolar, bring them together, they form adhesions between themselves, but they're still mesenchymal. I put them on a substrate. If the integrins in the cell like to stick to the substrate, the cells automatically polarize. Not very much biology in here, but it's actually not bad for a lot of the things that we do. You can always link it to a subcellular model for reaction kinetics. Drosophila wing, you have hairs that are oriented due to planar polarity. There's a big debate about how that planar polarity is transmitted from the membrane, from the boundaries in the wing through the wing. If you interfere with the planar polarity pathway, you get particular types of disorganization of these fibers. If you interfere with the boundary polarization, you get different kinds of disorganization. There are many models. Giulio worked on strut and strut cascade model. Has a fairly complex set of receptors, membrane-bound receptors, and signaling. I'm not going to go into the details here, which we represent in this simple way here. And here we have a boundary. That boundary is inducing planar polarity in these first cells. Those first cells then induce planar polarity, and you see the organization of the polarity propagating. It's actually not so easy to get that to work. You can use quite a few of the standard published models for planar polarity induction, and you discover that the planar polarity decays exponentially as you go away from the surface, or that it slows down as you get far from the surface, and other things like that. And then, of course, you need some kind of metric, and in this case, I'm not going to have time to compare it to experiment, but you can define an order parameter, a nice physicsy order parameter, and you can calculate the order parameter as a function of time, and it increases linearly, and that's basically what you see in the experiment. Most of the other models have some other time dependence of the order parameter and don't agree with the experiment. Let's now do something a little bit more complicated. Also Giulio, Bob Bacalhau, clinician researcher. Polycystic kidney disease, everybody's favorite. Before lunch. Polycystic kidney disease. You die when your kidney gets to be the size of a watermelon. No treatment except kidney transplant. Slow disease. Interesting because while it's a tumor-like disease, it never develops into a cancer. one of those very rare diseases that is essentially 100% dependent by mutation in one of two genes, very rare. Unfortunately, we don't know very well what those genes do. Kidney has a nice tubular structure. In polycystic kidney disease, the kidney grows cysts, and those cysts grow cysts on the cysts and cysts on the cysts on the cysts. And your kidney just keeps growing. And Bob and company discovered that if you intake a culture of normal human kidney cells, put them in a petri dish, they organize into tubules, which is normal. And then if you inject one cell, this is after a lot of gene trolling, with the adhesion molecule cadherin 8, you get the phenotype of polycystic kidney disease. And that's interesting because it winds up that the molecules PKD1 and PKD2 associate with cadherins. And so one of the ways that they may be functioning is to mediate contact inhibition of proliferation of cells through cadherin binding. All right. Here's the experiment. Here are the tubules of, these are actually primary human kidney cells. You transform a cell in green, and you'll notice that wherever you have green, it's formed a little cyst, and basically there is no green in the tubules. So we build a little bit of model. These are now polar cells, self-organized polar cells that make a tube. And now we say we're going to change the adhesion molecules in one cell and see what happens. And there are two ways that an adhesion molecule could function. The normal complement of adhesion molecules is still present in these cells, but there is an additional one. The additional one is quite interesting because it's a normal molecule in juvenile kidney that gets turned off when the organ stops growing. It could be interfering with the adhesion directly, or it could be interfering with the transduction of contact inhibition signaling, or it could be interfering with both, and you can model those independently. And here is a model where it interferes with adhesion, but not signal transduction, and you get a cyst. You notice the cyst grows out as a little circle. And you can also interfere with signal transduction without adhesion, and you also get cysts, but they spread within the plane of the tubule. And you can actually, I'm not going to show the data, you can actually quantify the morphology of the cysts compared to those in the simulations. And we then went back based on that and actually tried to measure semi-quantitatively the adhesivity. And sure enough, the adhesion of these CAD8 expressing cells is lower than of wild-type cells. And that actually gives you a mechanism, and then potentially you could think about drug targets for that mechanism, which we're doing. The last thing I want to talk about was age-related macular degeneration. I'm going to run out of time. Boy, am I going to run out of time. As you get older, there's the possibility of the blood vessels behind your retina invading the retina. Not a pleasant disease. It's essentially a disease of older people, but in young adults, if you get acute inflammation of the eye or septic shock in the whole body, you sometimes get invasion by blood vessels. This invasion looks a little bit like neovascularization in tumors. Interesting thing is that in older adults and young adults, the locus, the position where the invasion happens, is different. The standard theory has been that there's excess growth factor in the eye, or that there are holes in the membrane supporting the retina. There are excellent reasons to believe that those don't work, but there are more than 5,000 papers a year published on the hypothesis that that's what's going on. And there are hundreds to thousands of drugs in development trying to treat that idea. And so we build a little model of the system, and you run it, And here is a cell, endothelial cell that's been activated by high levels of VEGF. It's made a hole in Brooks membrane, and it's trying to invade the retina, and it doesn't. And then we had a little bit of a piece of luck due to FedEx, which I'm not going to tell the story of today. Asked me about it over lunch. But as you get older, gunk accumulates on the membrane that supports the retina. And if you want to glue something to a surface, you don't coat it with soap before you glue it. And so that's why when you're old and you fall over, you get detached retinas easily. Inflammation reduces the lateral adhesion between cells within pigmented retinal epithelial layer. Mechanical trauma tends to reduce adhesion between the two layers at the bottom of the retina. And you can now put in reduced adhesion between the pigmented retinal epithelium and the supporting membrane, and now that same cell is able to invade. And actually, interestingly enough, the time scales of invasion are about right in this simulation. We actually did validate the times pretty carefully in this. And you get this spreading, which is typical of the disease. You can then say that the disease is heterogeneous. Same initial conditions, 10 runs, 10 outcomes. That means that it's not a determining thing, it's a risk thing. That's known experimentally. And you can build what are called risk maps. Start with a normal tie with normal adhesion of the three kinds. You don't get the disease. If you weaken one, you have a certain probability of getting it. Weaken another, weaken a third. Each one has a different probability through a different mechanism. In the case of the particular disease that we were talking about, which is called sub-RPE CNV, we find that adhesion between Brooks membrane, the sub-underground membrane, and RPE is the critical thing. RPE adhesion, which is reduced in inflammation, doesn't make much difference. RPE photoreceptor adhesion doesn't make much difference. So mechanical trauma shouldn't cause this problem. Inflammation shouldn't cause this problem. Only aging. And that's what's observed clinically. Okay? And you can play games. Think about the inflammation case. You can show that the invasion happens in the right place. And you can make it go a little further and ask questions about how the membrane breaks down as it's mechanically disrupted and interferes with blood flow and so on. Okay. So what can you do? What's the lesson behind all this? This is for the students. It's really for everybody, but I won't presume to tell everybody what to do. I can presume to suggest to the students that they might think about this. And then you can choose not to do it. But if you're not going to do it, think about it. Separate your models from your simulations. That is, write a formal biological model, no numbers, that outlines the entire biological structure of what you're going to be describing. And that is after you've made all the simplifications and everything else. That looks like a pathway diagram in a textbook with a little bit more detail. Then write your math. And then if you're going to turn it into a simulation, write your simulation. Keep those three stages distinct and keep the components distinct. Follow best practice workflows for model development, and there are standard best practice workflows. If you're developing models, keep the components separable. Ideally, make sure that they run independently and can be validated independently of each other. Annotate. We all know about that, but we don't do it. Where do we need to go to make this into a serious, in quotation marks, field? Standards for model and simulation components. That has to come from a mixture of community and top-down stuff. Start filling in the gaps in tools. We've been focusing too much on solvers. That's not where the holes are. Design tools and methods that work together. Use standards and standard components when they exist. If there's something that's good enough, use it unless there's a compelling reason to do better. Doesn't have to. It's better to use a standard that's good enough than a non-standard that's 50% better. Focus on users and usability. Be cooperative. Think about product development models and interchange. Think about open source software movement. And for the applied mathematicians in the house, we really have a problem describing extracellular matrix. Complex fiber architectures that are inhomogeneous and anisotropic and have long-range mechanical interactions are essentially impossible to solve numerically. And if there's one thing we need, it's not a better fluid dynamics solver, there are plenty of those. It's not even a better membrane solver, although we could use better ones of those. It is a better ECM mesh solver. And also better tools for doing what we would call immersed boundary coupling between 1D, 2D, and 3D levels. That's why I found what you were talking about so impressive today, because of this focus on coupling 2D to 3D. Have to get 1D in there too. Because biology has fibers, surfaces, volumes, and it has meshes, and meshes are a pain. I wish Fred McIntosh were here. He's the Mr. Mesh, but anyway. Okay. So, moral. People were saying, but this is an impossibility. There's no support for it. There's no institutional commitment to it, and so on. Things are changing. At NIH, there's a group called IMAG that was led by one person, Grace Peng, in imaging. She has a pretty decent budget of $500, $600 million a year to put into this stuff. We've organized over the years 3 workshops at NIH for program directors to explain to them why this stuff is important. The world does listen, even if it feels like you're talking at a wall all the time. So don't give up. Don't give up. But absolutely, and this is perhaps the single thing that I think is most critical, mathematicians, physicists tend to want to go off and beaver away on their own projects. Biological problems are too hard for any one individual to solve everything. We have to give up some of our autonomy and work together. Otherwise, we're going to wind up with another euro. The people don't learn from history. If you look at the European Constitution now, it's almost identical to the Articles of Confederation of the United States. How many people have heard of the Articles of Confederation of the United States? 3, 4. Okay. After the independence of the United States in the Revolutionary War, the individual states did not want to give up their sovereignty, and they organized a loose organization with the same economic organization as the EU, the same structure of voting and everything else, and it lasted for 8 years. And there was a complete social and economic breakdown that led the states to finally say, we're going to give up sovereignty, pool everything, and have a central government. I'm not saying we have to go there, but if we don't hang together, by heavens, we'll hang separately. And by the way, if you look on Wikipedia, the source of all knowledge, that's either by Benjamin Franklin or Jared Sparks or Richard Penn or Frederick Reynolds or Dryden, the playwright, or Carter Braxton or someone else. I think that emotion and words have been expressed many times. Okay, it's an intimidating list, but think about the payoffs. Every time you use your cell phone to navigate, think about the importance of that. GIS can serve as a template for almost everything we do. Almost all the problems we address are in geographical information systems. If we can make it work, rapid scientific progress, identification of error, reduced duplication, models as equal partners of experiment, and models that work in a clinical and regulatory setting, models that make a difference, not just to us and our publication records and our promotions, but make a difference to everybody. If we can help keep 20% of people from going blind, or help people overcome cancers with better dosing schedules, or treat the 2% of people a year who are going to die from polycystic kidney disease, and a model makes a significant contribution, that's important. And ultimately, I guess I'm an engineer. I believe that while the abstract science is beautiful, and it's wonderful to do poetry, what gives value ultimately to us and justification is the ability to make a difference. And if we want to make a difference, we have to change the way we do things. If you're interested in any of this, we're giving our annual CompuCell course and System Biology Workbench course August 9th, August 15th in Bloomington. Probably too late for people this year, but if you have a student or something that wants to come, we'd be delighted to have you. We've been giving that course now for about 8 or 9 years. We've given it a couple of times in different places. We gave it in Spain once. We've given it in Germany. We've given it twice in Leiden. We've given it in China and Taiwan. It's fun. People can come and kibitz, try out our systems, tell us what's wrong, go away and say it'll never use it again. That's fine. But I think, I hope that's a sort of a different perspective. It wasn't what I was planning and talking about, but hearing the talks, the things I was going to talk about were all said, so I thought I'd talk about this instead. Thank you.
I think that was a stimulating talk. I'm sure there's lots of questions. I've got lots of questions. And
Does anybody have darts? You can all see how good your dart practice is with me.
I thought it was a great talk, yes.
So if you go to the later, more complex applications you use to illustrate CompuCell 3D,
Yes.
They're very good, but they start to look a lot like the typical modeling that we have been seeing at this workshop. That is, you start with biological concepts, assumptions, you translate that into a mathematical framework, you implement it, right? Now, can you take then CompuCell and go back to the list of criticisms you had on the current mode of operation? For instance, the lack of modularity, the lack of reproducibility, and demonstrate how you can use a framework like CompuCell to address those issues.
Well, there are a couple of problems. One of them is that open source modeling, it has a bootstrapping problem. The effort to develop open source software is about 10 times the effort to develop closed source software to do the same thing. That means that until you have 10 groups that are developing software to the same framework, you lose. While there are dozens of groups around the world that use CompuCell to build models, and there are large numbers of publications using it, the number of groups that have actually developed core software for CompuCell is fairly small. Partly because of the social and financial things that you were talking about. We would love to be able to integrate other cell representations into CompuCell. We'd love to have somebody's cellular automaton representation like the ones that Mark Chaplin does. We'd love to have center models like Dirk Drees does. We'd love to have immersed boundary and finite element models. We'd love to have level set models and phase field models. On the other hand, we're not experts in any of those techniques. It makes a lot more sense for the people who develop the engines to work with us to wrap those engines, rather than for us to try to redevelop the core code, in which case we may not understand it. The group we've worked with most closely is Herbert Sauro, who's one of the core developers of SBML, to integrate the subcellular reaction kinetic network modeling with CompuCell. And so those were two packages that were initially distinct that have now been merged together and function more or less seamlessly together. And SBML lets you choose Gillespie stochastic solvers or ODE deterministic solvers. But ideally, yes, you want to be able to change your diffusion equation solvers. We have a bunch of diffusion equation solvers. Once we get Phoenix in, you'll have a lot more. But we're working on that.
More questions?
This is somewhat indirect, but I have some concern about producing these very large-scale packages into which people can just plug a few things without having a pretty good understanding of what the limitations of all the things that went into it are. I mean, you held MATLAB up as a nice example, even though it's commercial, but there are examples where people use that to solve simple differential equations which then oscillated around zero, even though the variables they were describing are concentrations, and they published these results. All they did is they used Euler step in MATLAB and...
That's a biophysical journal. It's still being cited.
Yeah, it's in the Biophysical Journal of all places. But my point is, there has to be some effort throughout the community as well to get a better qualitative understanding of what to expect in different contexts. So you don't generate garbage like that. And that is very hard to address, because the minute you say, I've got a package that's going to solve all your problems, people are going to stop thinking about what are the limitations.
If people knew how many people would die every year from automobile accidents, automobiles would never be licensed. Now, that's not an excuse. I agree with you completely. And that's why a statement like biologists don't need to know any mathematics is such a devastating one. It shows real ignorance. However, it's not, in a sense, the tool's fault. You do want the tool to come with warnings about situations in which you know it may misbehave. I think of Mathematica. The first time I used Mathematica, I typed in a Turing equation, and it happily threw me a solution, which made no sense at all, because somehow the integrator it had chosen to solve the Turing equation was unstable, and you had no way of knowing what integrator it used, what time steps it was using, or anything else. And it didn't give you any warning. It gave you a beautiful picture that was totally meaningless.
But that's because you understood what to expect. It was meaningless. If you didn't, you might have taken that as truth and gone from there.
Yes. So one of the ways one can do that is by giving guidelines for parameter values, for typical output. And that's one reason that training material is very important. In other words, the problem is, of course, you can't keep people from not reading the training material. How many of us have read the instruction manual for the software we use? Most of us haven't. You just use it, and then you see what happens. I agree with you completely, but that's not a reason not to develop the tools.
Oh, I said it was
It's part of this social transformation that needs to happen.
This is reproducibility, isn't it? I mean, you've got
Right.
different. MATLAB is just one.
Right.
Actually, in your big diagram, in your mathematical model of how to do science, within the context of useful biology, which is essentially what you've kind of given you could say it's a computational model. You could also say it's a mathematical model of how to do science. MATLAB would just be one small component of that.
Right.
And this question that you're talking about was addressed with respect to reproducibility.
Exactly. So, yeah, I would just say, I mean, ideally, if you have one of these packages that's well designed, one of the standard workflow things is you take whatever solver you've used for element X, you replace it with a different solver, and see if you get the same answer. Now, of course, they could both be failing in the same way, but it's much less likely that they're going to fail in the same way. And if they give you a fundamentally different answer, then you have a warning that it's an artifact. I mean, airplanes do this. They have software for controlling the flaps that's written on different computers in different languages by different groups, and they vote. Now, sometimes it winds up they somehow pick the same algorithm that has the same mistake, and then you're toast because they both make the same mistake. But the goal is that the more independence you have, the more you hope that what you're getting is a representation of the fundamental model and not of the instantiation. But yes, but again, until you have the tools that allow you to do this, at the moment, if somebody publishes a model using PDE solver X, and I want to solve it with PDE solver Y, I have to re-implement everything from scratch. Yeah.
In your grand scheme, the grand scheme would mean that you'd have to have vast increase in the number of people, vast increase in the number of groups, and somehow they would have to cooperate. And I guess this is the question of whether that can be done top down or whether it might just emerges bottom up from the behavior of all these groups, whether something will, the structure that you want, whether it would emerge or whether it's got to be implemented, it's got to be imposed from the top.
And there is an excellent example for the system that you are talking about, and this is the standard example in computational chemistry. It's called the molecular dynamics method.
So, molecular dynamics, yes, of course.
Molecular dynamics, I mean, there are maybe 2 or 3 now main integrators that are on the market, and there are millions of people using molecular dynamics all the time to generate in, I will not say the percentage because I don't really want to offend anybody, but in a large percentage garbage. In some smaller percentage, it is a very good and good quality work. And this good quality work is actually done by experts in molecular dynamics, where the rest is very often somewhat periphery, yet the conceptual framework of molecular dynamics is very simple. It is Newton equations plus the force field. And I think here the problem is, well, I fully understand where you're going and that you need to do that. Here the conceptual framework is incredibly complicated. And even
The physics is
physics is not known, so
Yes.
that's why I find it, but it's not to do it, it's just that the obstacle is much larger.
Right?
Yeah, this is also something I would like to mention. You say bioinformatics is doing better than biomathematics. True. But there are also examples in mathematics where mathematics is sort of successful along the way you describe, and that would be linear optimization, for instance. Yet you have tons of pre-processing tools which pre-process your variables, scale them in an appropriate way, and eventually there comes a solver which can be replaced, you get some results, and then you have a full-scale post-processing tools, and you have standardized standards for describing your problem, and so on. And there everything works successfully. As soon as you go to nonlinear optimization,
Can't do it.
nothing is there like this. And I think what is important is to identify problem classes where you can standardize ways of how to approach them, how to define a suitable workflow which works for biologists and also for mathematics side. I think this is the biggest problem. What is discussed here is so diverse and non-linear maybe.
But sometimes the nonlinearity works in your favor in the sense that one of the lessons of modern complexity theory is that a lot of times, provided that the interactions are roughly right, the detailed behaviors of the agents aren't that important. And so if what you're interested in are the higher level emergent properties, you can sweep a lot under the rug. No guarantees it works. These are all phenomenological models. They all have to come back to experiment for validation. But you can design experiments much more intelligently. You can interpret experiments much more intelligently. And I think the example, perhaps, that you give is a good one. CAD-CAM is a good one. I mean, when you're doing CAD design, there's almost limitless range of things that those tools are used for. And so they have to be incredibly flexible. Now, that doesn't mean that sometimes you run into a situation, my God, where it doesn't work. It gives you the wrong answer. So you always have to build the model, physical model, to check. But you can do a lot computationally now. And the goal is to encapsulate more and more. In the same way with the concepts, you have a concept of a cell we can define now. We have a concept of something like cell growth or cell division we can maybe define now. But you always have the thing which the purists in this field would dislike, which is you have a black box that says you can put anything here. You write your program. That little thing you've written is not shareable. It's not, maybe not be modular in a technical sense. But the goal is that when you see the people doing the same box lots of times, and people agree on it, then you encapsulate it and incorporate it in the basic code. So you have a dynamic standard that allows growth, not a frozen one. No.
I think, I mean, I'd like to have a longer discussion on the issues which were raised by this talk, but I think that it's 12:35. So we should probably thank the speaker and people who've got questions and want to have an argument and etc. There's plenty of opportunity to do this.
I guess there isn't a talk this late afternoon, so I'll hang around and you can throw darts at me all afternoon.
Well, I would suggest maybe we start the last talk 15 minutes later.